Welcome to KT Study Zone. In this video series, we are discussing about the chemical basis of life. This is part 8 and we're going to talk about the practicals. Specifically, these are the practicals to be discussed. The test to identify carbohydrates, test to identify proteins, the test to identify lipids, the test to identify water as a constituent in food, the test to identify the presence of carbon in biomolecules, and the test to identify nitrogen in biomolecules. First, iodine test, which is used to identify starch. Well, in each test, I will discuss about the method, the observation, and the conclusion we can come through the test. Method. Small amount of food, for example rice, is obtained and grind well with water. Then a drop of iodine solution is added. We can observe purplish or dark blue color will appear. Conclusion. The sample contains starch. Next is the Benedict test. We can use this test to identify reducing sugars like glucose and non-reducing sugars like sucrose through a series of color changes, as you see here. So, let's study about it. First, we will see the Benedict test for glucose. Method. A solution of glucose is obtained into a test tube. Then, a few drops of the Benedict solution is added to the solution. Then the solution is immersed in a water bath and heated. Observation. We can observe a color change as below, from blue, green, green-yellow, orange to a brick red precipitate. Well, the conclusion is the sample contains a reducing sugar. Next, we will see the Benedict test for sucrose. Method. A solution of sucrose is added into a test tube. Then, a few drops of Benedict solution is added. The solution is immersed in a water bath and heated. Observation 1. There is no color change. Then, a few drops of dilute sulfuric acid is added to a freshly prepared solution and it is heated in a water bath. Next, few drops of Benedict solution is added to it. Observation 2. Can observe the color changes as below, from blue, green, green-yellow, orange, and to a brick red precipitate. So this concludes the sample contains a non-reducing sugar. This is because sucrose, which is a non-reducing sugar, usually does not answer the Benedict test. But when sucrose is broken down into glucose plus fructose using the sulfuric acid, a reducing sugar is formed. Then it answers the Benedict test. Next is Biuret test. Method. A solution made by grinding dal or egg white is obtained into a test tube. Then, an extra amount of sodium hydroxide and a few drops of copper sulfate is added into it. Observation. The solution will turn into a dark purple color. Well, we can conclude the sample contains proteins. Alright, now Sudan III test. Method. Some amount of gingerly oil or coconut oil is added into a test tube. Then Sudan III reagent is added into it. Observation. Red fat globules will appear. Conclusion. Sample contains lipids. Identification of water as a constituent in food. Method. Grind or crush meat, eggshell, and leaves in their dried form separately. Put them separately into crucibles and heat them. During heating, hold a glass sheet above the crucible. Use anhydrous cobalt chloride or copper sulfate to identify whether the liquid drops on the glass is water. So, the observation is blue-colored anhydrous cobalt chloride turns to pink and white-colored anhydrous copper sulfate turns to blue. Conclusion. The sample contains water. Identification of presence of carbon in biomolecules. Method. Make pulps by crushing some spinach stems and pieces of fish separately. Put them separately into crucibles and heat well. Then, the final residue obtained should be rubbed against a white paper. Observation: lines drawn due to coal is observed, which concludes the sample contains carbon. Finally, identification of presence of nitrogen in biomolecules. Well, for this, Biuret test is used because it confirms the presence of proteins in food, and as nitrogen is a constituent of proteins, the sample contains nitrogen. That's it. So, thanks for watching. Don't forget to like, comment, and subscribe.